So welcome everyone. Thank you for participating in this webinar. It is about endocrine cell isolation, and I have here with me Dr. Phil Streeter and Pamela Canaday from the Oregon Health Science University. My name is Nirja Patel, and I'm currently managing the diabetes and obesity portfolio of products here at ALPCO. So Dr. Phil Streeter is the director of the Center's Monoclonal Antibody and Flow Core Facility. His research areas have included tissue engineering, cancer diagnostic tools, as well as beta cell replacement therapies for the treatment of diabetes. And Pam has been the main flow operator at the core facility there. Just a little snippet on ALPCO. We are a medical diagnostics company that specialize in distributing and developing ELISA assays. In about 2013, we started we introduced our flow cytometry portfolio of products, which included conjugated monoclonal antibodies for flow. And so this webinar is essentially, the goal is to present with you some of the technicalities that go into flow cytometry, specifically how Phil has used some of ALPCO's pancreatic antibodies for some of the work that he's done at his core facility. So without further ado, I am going to hand it over to Phil and Pam. And I would encourage you to come up with questions. However, we're going to wait to answer any of the questions till the end. We'll have a little Q&A session at the end. But if you do have any questions, again, please feel free to type them in the little chat function or box at the bottom right corner of your panel. So having said that, here we go. Phil and Pam, take it over.
Thank you very much for giving us the chance to describe some of these reagents today. The discussion will be divided into a couple parts. One where I'll start out by describing a couple of reagents and actually how we prepare cells for flow cytometry. And then Pam is actually our expert at sorting these cells and working with the antibodies that way. And she probably has more experience than anyone else in the world, maybe, with sorting beta cells. So we're really excited to have the opportunity to share this to you today, or to share this with you today. This slide shows is really the two antibodies that we'll be talking about today. And what I wanted to emphasize before we get started is that what we look at in our laboratory are dissociated islets, most of them from normal donors. Sometimes we get donors that have, say, type 1 or type 2 diabetes, but what we'll talk about today is our observations with normal donors. And really what I wanted to emphasize here is that these are the type of results I think you should expect from, again, normal islets. You may see something quite different if you're looking at cells generated or islet-like cells generated in culture. And that may be because their transcriptomes or proteomes may be very different than, or maybe somewhat different than cells derived from islets. So I just wanted to kind of let everyone be aware of that. I actually hope someday we can actually have someone look at these reagents using, say, beta-like cells generated in cultures and then transplanted to xenogenic animals and actually look at the outcome from those transplants. That would be quite exciting. So again, what I wanted to talk about first, and then I'll hand it over to Pam, is really how we prepare the cells for flow cytometry. And again, we're starting with human islet samples. We've obtained these from commercial and non-commercial sources. What I wanted to acknowledge today is that we actually get most of our islets from the Integrated Islet Distribution Program. We really are very happy with the islets we received from them. They've been very high quality. But those islets are usually cultured overnight prior to shipment to us. And then upon receipt, we actually either use them immediately, we try to do that, or we sometimes have held them overnight before using them. But what we do after, and when we're trying to, or when we're dissociating those, is initially spin those down at 300 G for approximately 4 minutes. We discard the media, and then we disperse the cells into single-cell suspensions using high-clone trypsin, and that's pre-warmed here. I'm emphasizing this because, again, this is the enzyme we use for these dissociations, and one could see different results if, for example, a different source of enzyme is used or a different type of enzyme is used, and or if that enzyme is, say, left on the cells for a longer period of time, you might actually lose expression of or lose the markers that you're trying to detect with the different reagents. But what we use, again, is high-clone trypsin. We use 3 mils of that trypsin in each tube and incubate that for 10 minutes. Usually the dissociation procedure involves anywhere from, say, 2,000 to 5,000 islets. That's a good general rule there. And then the cells are gently dispersed by pipetting during that 10-minute incubation period. About every 3 minutes, they're gently pipetted about 10 times to help dissociate the islets. And again, just wanted to emphasize that over-incubation with the trypsin can result in loss of some of the markers from the cell surface. This dispersed cells, we try to get them to the point where there's not many clumps, but there's always something there. The dispersed cells are then passed through strainers of 35 to 40 microns, and then those dispersed cells are rinsed with media again, and then with 2% fetal bovine serum. That's used to collect the cells that are the residual cells in the tube, and then actually that media is also added to the dispersed cells. And then cells are centrifuged and brought up in actually the CMRL media, again with 2% fetal bovine serum for storage prior to staining. We also add, we haven't always done this, but we also at times add trypsin and chymotrypsin inhibitor to the cell suspension to actually inhibit any residual enzyme in those tubes. So actually with that, then the cells are counted and then stained for the flow cytometric application. And so this time I'm going to actually, and some of the methods for staining, actually ALPCO already has literature on that, so I wasn't going to cover that here. But from there, actually, I think I'll at this point hand off to Pam, and she'll describe actually how she works with these cells once they're prepared for sorting.
Hello. So first of all, the thing that I want to stress is that you need to optimize your flow cytometer. We have an influx, and we use a 100 micron nozzle with 15 psi sheath pressure, but it's really important that you look at your sorter and play around with different settings and different nozzle sizes and empirically choose what is going to work best for the cells in your hands. The blood and bone marrow tends to be sorted under higher pressure and with a smaller nozzle, and those can just shred the islet cells. So if you're doing a typical 70 micron 45 psi sort, you're not going to end up with very many viable cells at the end. So larger nozzle and lower pressure. The other thing that we find tremendously helpful is standardizing our forward and side scatter plots, and we do this by running 10 micron beads. And if you see where the little green box is here, 10 micron flow checks will make a line on forward and side scatter, and we put that line right about in this place every time we do a sort with islets, and that way we get very reproducible forward and side scatter profiles, and it makes backgating really easy and wonderful. When you put these on, it's noteworthy that the side scatter voltage is going to be considerably lower than it would be if you were running hematopoietic cells. Another really important thing before we even get to the antibodies is doublet discrimination. If you want a highly pure sort, you need to have a really tight doublet discriminator gate. On the influx, we use pulse width. On other sorters, you'll use forward scatter width. It's, if you don't need such a pure sort, if you're just going for enrichment, then it's not quite as vital. But you're likely to get like endocrine cell, other cells will stick to beta cells, for example, and anything sticking to an alpha cell will look like an alpha cell. So that's just an important feature.
Okay, this is Phil again. I just wanted to jump in. What Pam's going to describe today is really how we achieve very highly enriched beta cells or alpha cells. Again, if someone is less interested in a high level of purity, you can actually, give a little bit back on some of these parameters that you'll describe.
Yeah. So the first gate that we do after the forward and side scatter and singlets gate is live dead. In this graph we have HPa3 and PE. We don't compensate because it tends to distort the PE signal, and it distorts it enough that it will blur the line between the HPa3 negative and positive. So, and it's very clear which ones are PI positive and which ones are PE positive. So you don't really need compensation in order to distinguish between those. So first we'll look at beta cells and how to gate on the beta cells, and after that we'll look at the alpha cells. Beta cells in this case are defined as the population that's HPi2 positive and HPa3 negative. So this is where the uncompensated PE shows a clear distinction between things that are PE positive and PE negative. So the negative is showing up along the diagonal. The high autofluorescence cells here are actually the ones that this population here includes the beta cells. And, sorry, lost my train of thought. But it's if you were to just draw a straight gate going down in one place, if you were to draw it here, you would exclude these HPa3 positive cells, but you would also be excluding beta cells. And if you were to draw it here, then you would be including a number of HPa3 positive cells. So if you just look at it, HPa3 versus HPi2, thank you, then the boundary isn't as clear. So it's important to gate on the HPa3 negative first. And before I forget, don't make the mistake, please, of gating on histograms, because if you do that, you are going to, it's very likely that you're going to be including a lot of events that are not beta cells in your sort. And this is just an example of the green dots are HPa3 negative. I find it's helpful to show this overlapping. There are pink dots that might normally be included if you gated on HPi2 versus HPa3. So this is just another illustration of why it's so important to gate on the HPa3 negative first. And then after that, you can move on to gate on the HPi2 positive. You'll notice here that it mentions HIC12D12. HIC12D12 was just the original name of the HPa3. So sorry about that. Yeah, and after you gate on the HPi2 negatives, it's really easy to tell which ones are the HPi2 positives. They're higher in side scatter, you can see here, and it's what I forgot to mention in the forward and side scatter slide is that we make our forward and side scatter gate off-scale high to include the off-scale high events on side scatter, because since beta cells are very granular, they tend to have significantly higher side scatter. And if you are interested in also sorting alpha cells, then you would use the same live dead gate and just gate on the double positives. For a really pure population, it's really important to make a tight gate, because otherwise you end up getting other endocrine cells included in your sort. So I think that sums it up.
Okay. Yeah, I just wanted to reemphasize the last point that Pam was making, and that is that you do want to avoid cells that are down, say, in this area here on the plot, Again, there are different endocrine cells, and also this kind of mix of cells that are between these two populations. You would like to make a tight gate around the alpha cells there, and then you will actually achieve a very high enrichment of those alpha cells. So I guess from there, we maybe hand this back and try to address some questions.
So the first question is, are they still functional after sorting?
That's a great question, and it's actually something we are in the process of doing currently. We have a manuscript that's been submitted where that information has been requested. That's not something we've routinely done in the lab, but we've recently brought the technology in. So I apologize, I don't have the answer for that today, but we're actually trying to do that at this time. I don't know why they wouldn't be functional, because they certainly are viable after these sorts. But one consideration might be that if you require more than one endocrine cell type to see function, that having them separated into discrete populations might make that more difficult.
All right. The next question is, what is the overlap between insulin-positive cells by cytoplasmic staining and your surface surrogate markers?
Let's see. With the way Pam has illustrated, I don't have that slide or a slide of that right now, but with the way Pam has illustrated these gating properties or the strategies, then there's actually a very high correlation between, say, insulin presence within the cells and the beta cells I identified with those markers. If you actually look back at the stain or the staining with the two antibodies, where if you look, say, between those two major populations, that's where you can actually see some alpha cells. Say if you stain with a glucagon-specific antibody, that you would actually not be collecting here. And again, the reason Pam showed you the gating strategy we use is to achieve a high enrichment of alpha cells. So there is a little loss of alpha cells as well as beta cells, but we're sacrificing a few of those cells again to get the high level of enrichment.
Okay. Another question was if these...
Sorry.
I'm sorry.
Yeah, I actually apologize. I use the word enrichment. Again, what we're trying to do is to achieve a high level of purity. And then using these strategies, again, the hormone levels or hormone expression correlates quite nicely with these populations.
Okay. The next question was, can we get the presentation slides? And the answer is yes, they will be made available. They will be emailed to you after this webinar is over. What you're seeing on the slide right now is my contact information. If you have any questions, please feel free to reach out to me directly. And it doesn't look like we have, we have one more question. What staining conditions are used for the antibodies?
Okay. The staining conditions, what we routinely use is actually for a couple, I think for these reagents or these plots you saw today, maybe Pam can help me with this. I think these were two-stage stains.
Yes.
So we use secondary antibodies or unconjugated primary antibodies, and the secondary antibodies were conjugated. The staining conditions I prefer are to actually do the stains on ice for roughly 30 minutes with each reagent, and then washing them between, say, after staining with the primary antibodies before secondaries. I usually would use cold buffers for that type of manipulation. The reason we're able to actually detect both of these reagents or both of these antibodies in different colors using secondary antibodies is that one of the reagents is an IgM and the other is an IgG. And so what's done here is the secondary reagents are specific for mouse IgM versus mouse IgG. And actually, ALPCO has the information for you on what those secondary reagents are as well, or the providers of those reagents.
Yes. Our next question is, do you have antibodies to sort mouse islet cells? Unfortunately, at this point in time, we do not, but we're looking into it. It's on our radar. The next question is, do longer incubations improve separation?
I would actually say I don't believe so, and the reason is I think with the conditions that are set up here, we're really seeing pretty saturating staining at these conditions. So I would say no. Pam, do you have anything?
I think from my experience, longer staining just increases the background.
Yeah, so I think again I'd recommend the staining with each reagent at roughly 25-30 minutes, that time frame.
Okay, our next question is, can we have the staining protocol?
Absolutely. Actually, ALPCO has that available, I believe. Is that true? We've written up a staining procedure for these reagents.
Yes, so that will be made available. Yes.
And any other questions, we can address anything that is missing or if it's unclear.
That's right. The next question is, how well do these antibodies stand to fixation if not using for sorting?
That's a good question. What we routinely stain with is acetone for, say, immunohistochemical purposes. So if you're interested in actually doing immunohistochemistry, we know that the reagents will work with acetone. Some of them may work with, say, a paraformaldehyde or formalin fixation, but we really haven't validated that. What I can say is if you're doing a flow cytometry, if you just wanted to do analytical work, you could certainly stain with these reagents, then again go through a washing procedure afterwards, and stain with some kind of a FAX fixative, which is usually say paraformaldehyde at 2%. But they can be fixed with paraformaldehyde after staining. But I would not recommend fixation before staining. We haven't validated that anyway.
Okay.
Is that clear?
Yeah, I mean, I'm sure to hope so. Are there any other questions from the group? I believe not. Having said that, thank you, Phil, thank you, Pam, and thank you everyone for participating. Once again, these slides will be made available to you. And should you have any questions, please feel free to reach out to me directly. My contact information is on the screen, both my email as well as my phone. Thanks, everyone.